one thing I want us to focus on is how you're able to take the DNA and you transcribe DNA to form RNA, specifically mRNA. And then what was the outcome of that mRNA? Where did we take the mRNA? So mRNA exits through a nucleus, or through a pore, a nuclear pore. And you said it goes to next the ribosome, right? Now what's going to happen at the ribosome that's pivotal? So this is the location of translation, which is basically when you read the mRNA to form the protein. Correct? Yes.
On the third bullet on the nucleus says mRNA
through a pore.
Oh, okay.
So you read the mRNA to form a protein, right? Now the ribosomes could be found in two different locations. Sometimes they are free, and sometimes they are attached. What would they be free, or why the term free, why the term attached? What are we going with here?
Free is in the cytosol,
Okay.
attached is to the ER.
Now, we're still going to be producing a protein on this ribosome, but there's two different locations of ribosomes. So what do you suspect the ultimate use for a protein created in the cytosol is versus that which is created on the ER?
The cytosol is for inside the cell.
Okay,
ER
so
is for out of the cell.
So in this case, the protein is destined for cell. And then in the case of the ER, the protein must be destined for outside of the cell, right?
Isn't there a word for that? Like, it starts with S, and we're reading
Like secreted. Like secreted.
secretory.
Okay, like a secretory cell. So a secretory cell is a cell that can produce a lot of objects and then send those objects away. So secretory cells would have tons of rough ER because they're making proteins that are typically sent out of the cell for some other destination. Now, if we continue this logic on, what would be happening in your rough ER that's helping out these proteins? And we're getting to where we left off with yesterday.
Well, the ER, it like folds, protects the proteins.
Okay, so we've got some folding and protecting, thank you, mechanisms.
Facilitates transport.
Folding and protecting the proteins. Basically you're helping them, you're facilitating the protein survivability, right? So you're kind of guaranteeing that the protein is going to be okay if it spends some time in the rough ER being modified so that it could last outside of it, correct? So I'm sorry, what?
What is this?
Facilitating the protein survivability. So you're kind of helping ensure the protein could live in an environment that is not of that cell. Good? Okay. So moving on, the next stop would be, since I've gotten my protein good to go, the next stop would be, how can I get my protein out of this cell? Because it's been made from an mRNA transcript. We've got it modified in the rough ER, so the next stop would be your Golgi, which is going to be the location in which we can start working on getting this protein out of the cell and it going in a correct direction. So, the Golgi, or sometimes called the Golgi apparatus, is going to have some important functions. And its major functions would include continued modification. It itself can manufacture carbs, fats, and nucleic acids. And lastly, and its most important feature, the feature that you're probably most familiar with, is how it is able to sort in packages, or sort in package. Bless you. Certain materials into transport vesicles. Now, one thing that we want to get an understanding with respect to vesicles is understanding what a vesicle is. First off, a vesicle is a storage facility, but it's one that can move, so it's a moving storage facility. And not only is it a moving storage facility, but another thing that it does is it's going to be important in allowing or fusing with the membrane, so it can fuse with the membrane. To release the contents. So what I want to look for and what I want us to think about is an understanding of how these work out. When you have, let's just say that this right here is going to show a picture of your membrane. So let's say that's the cell membrane right there. Let's say that this structure that I have drawn out here is the Golgi. As vesicles butt off, there's a vesicle that's butt off. It's got stuff in it that's important. Cool? The vesicle is going to travel towards the membrane. And as it gets to the membrane, it fuses with it. It fuses with the membrane. So that afterwards, the contents of your vesicle are released out because the membrane of the vesicle fused with the membrane of the cell, and now whatever contents you have are to the outside. Hopefully we understand that the contents or the bilayer that exists around a vesicle is the same bilayer that exists on the outside of a cell. So when these objects fuse, when the vesicle fuses with the membrane, then your contents of your vesicle are now just outside the cell. And this process of going out of the cell is reversible. I could have had something come into the cell and it would work the same direction, the same way. We okay with that description? Okay. Let's talk about a lysosome next. The lysosome is going to be the membrane-bound like compartment. It's got a double membrane. And what it does is it works to hydrolyze waste and other trash. Now, when we say hydrolyze, what does that mean, to hydrolyze? So that would be like a big like reaction of breakdown, right? And so that would make sense. This is like the thing that can recycle objects. And what it can hydrolyze is it can hydrolyze fats, proteins, nucleic acids, and carbs. So all of your four major groups are represented here. Now, a couple of things about this lysosome. First off, it's got a pH that's pretty low. pH is somewhere in the neighborhood between 2 and 3, I would suspect, which means it's high acidity. And also, the lysosome is something that can break open when a cell is told to commit suicide. This is known as programmed cell death.
What would happen, like, if there's a virus that infected it with suicide?
Could be because there's an infection in the cell. It could be because the cell has a mutation that's been recognized by its neighboring cells, and the neighboring cells pick up on this and send a message to that cell to go ahead and pop open its lysosome. And when that happens, you can imagine the contents of the lysosome are pretty, like, intense. They're pretty, like hydrolytic, which means they can break down a ton of stuff. Not only can they break down the trash that enters into a lysosome, but if the lysosome were ever to splice open, it would start to break down itself. The cell would ingest itself, and you'd be left with a bunch of little particles, a bunch of little carbs, fats, all that business afterwards, because the cell would be broken down, just like your stomach works out. Imagine if your stomach were to pop open. You could imagine that if your stomach is involved in having an acidic environment that can start to break down like food objects, like meat and whatnot, that if your stomach were to slice open, which should never happen, but if it were, that like hydrochloric acid, that acidic environment that is your stomach, would then be like inside your body cavity, which would start to break you down as well. Now, our case, it's not as extreme and very unlikely to happen, but for cells, this is an event that happens fairly often. Now, of course, once it does, there's no coming back. Once the cell has a lysosome that slices open, then the entire thing is over.
Now, what prevents that acid from, like, breaking down neighboring cells?
I think what it is that if you think about like the amount of space in between cells, there's a ton of water that's on the outside of cells, and it's much like saying that how come like if someone was to accidentally or on purpose cut like open a stomach of a human, what's stopping that human's like stomach from like destroying mankind? It's the fact that there's so much space in between us that acid would diffuse and would become such small and minute in concentration that it would hurt only the individual that it's sliced open in and not anyone else.
Okay.
So it's kind of that same logic. Cool. So the lysosome, important for recycling stuff, important for being involved in cell death, or at least programmed cell death. One thing I want to show you, I'll put this picture up real quick, is this picture right here. And one thing you can see in it, you can see like in this first picture right there, you have a vesicle or a vacuole that's coming into the cell, correct? And notice what fuses with it almost automatically the minute it pops in the cell. Lysosome. And the lysosome, you see how this membrane of the lysosome fuses with the membrane of the vacuole? Or we could say vesicle, because we can view the terms as interchangeable. Notice that what you have, you see how there's like this food particle that seems like very large in size? Notice that after the lysosome fuses with it, you're left with a bunch of little particles. Agreed? You see that mechanism, how that works out. Now, you'll not only see that the lysosome can fuse with like food particles that enter into the cell, but notice how a lysosome could fuse with maybe just a random mitochondria, or a vesicle that contains mitochondria, and maybe a peroxisome. You see how this lysosome is fusing with another vesicle? This lysosome could fuse with anything in the cell, and when it does, it's going to break down whatever it fuses with, and the contents of whatever it fuses with. Now, a good question would be, what's preventing this from like binding to the nucleus and breaking down all of my DNA at any given point? There's a lot of checks and balances in a cell, and that's one that's like always avoided. But just realize that the lysosome could not only like break down the stuff that comes into the cell, but it could break down the stuff that is a part of the cell if it's told to do so. Okay? Awesome.
Is there any like mutation that a cell can have that it's just lysosomes just go and
go crazy.
Yeah.
I would assume to say that if that ever happened, I would say that's that's probably a high level checks and balance system. So that would never happen to someone that's like the age of 60 and like their cells just like randomly lose that. And remember that if you have a mutation in one cell, that doesn't mean that every cell's mutated. It just means that one cell is.
Yeah.
So let's say you had one cell that went rogue and did that number where it mutated its lysosome interactions, and so now lysosomes were just destroying whatever it came in contact with. That's fine and all, but you have to keep in mind that, hello. You have to keep in mind that like that one cell goes bad. That's bad for that one cell, but that mutation was only in that one cell.
Okay. Yeah.
Thank you. Stephanie, it's for you. So we want to keep that in mind for sure. Cool. Now another thing could be, well, what if the only way mutations could be in every cell of your body would be if the mutation happened when you were one cell. Well, that would be like when you were first formed. And if the egg cell that was first to you did that, then that egg would never have survived, and you never would have made it to be here today, let alone to have even been born. Cool. The next part to discuss is the endomembrane system. Remember the endomembrane system, and your book went over this. What did the book list the endomembrane system as being?
A connection of inner membranes that separates the parts of the cell.
Exactly. So it was like a big system. That works together for one outcome. Do you remember like all the pieces that work together? It starts with a very central item. It's the nucleus. What else do we have? It was the rough ER was next. Then there were the vesicles. I'm sorry, the Golgi was next. And then you had vesicles. And maybe you had a lysosome as well. But if you look at this top piece right here and not kind of assume the vesicles not included in this list, so you have nucleus, rough ER, Golgi, vesicles. What are we seeing here? Like, what's the common like particle or macromolecule group that is all about these guys? Proteins. So you can guess that the endomembrane system has a heavy role, a huge role in protein production. Did I misspell roll?
No.
I did the first time, didn't I? I kept thinking honor roll. Isn't that ironic? If you're on honor roll, you can't spell roll. Ah, whatever. LOL, that means laugh out loud. Very good. Let's kind of get into a topic or a discussion about some energy organelles. I'm thinking about two. What are the two do you suspect I could be thinking about if I'm thinking about energy-based organelles? That's one of them. What's another one? Chloroplasts. Now, a lot of people are like, well, I don't think we should have this discussion because I don't have chloroplasts. And you're totally right, you don't. But the discussion was based on eukaryotic cells, and they're, in fact, if you were to survey all of eukaryotic cells on this planet, most of them actually would have chloroplasts. It's just unique that even though most of them have chloroplasts, we don't. So we kind of maybe sometimes accidentally limit our conversation to not including chloroplasts, but they certainly should be mentioned. So let's talk about what they're each doing. And what we'll see is that in like chapters 9 and 10, we explore these to a large degree. But for now, let's kind of just see where we got what we got with these guys. First off, about mitochondria, it's the site of cell respiration. The purpose of cell respiration is to generate ATP. Remember the formula is taking 6 CO2s, 6, nope, not that, reverse that, C6H12O6 plus 6 O2s to produce 6 CO2s and 6 waters. When you do that reaction, you also produce some free energy that can be used to generate some ATP. And I've drawn a little mitochondria there. A couple points to realize or recognize about it. First off, it's got two membranes. This is the outer membrane. This right here is the inner membrane. This space in between the two is called the inner membrane space. Why do we have this unique fold pattern here? More surface area. And that term that describes that is called the cristae. The middle part is called the mitochondrial matrix. On the very, very interior. So there's your pieces to your mitochondria.
Can I just ask a question?
Hmm?
Is the cristae the name
It's-
of the inner membrane?
It's
or-
the actual way that they're interfolded.
Okay.
So this is called an inner membrane. It's also referred to as cristae. It is truly an inner membrane that is inside of the outer membrane, but technically you would call this the cristae because it's not inner membrane that is right against the outer membrane. It's inner membrane that's been folded in.
Okay.
So cristae is a type of inner membrane that can exist.
What's the inside of the
Mitochondrial matrix. Now, if we come over here and we look at the chloroplast section, we'll see that chloroplasts are the site of photosynthesis. Which is a series of reactions that can generate ATP. In order to make sugar. Obviously, they contain a green pigment called chlorophyll. And the reaction that they're doing is the direct reversal is the one before, 6CO2 plus 6H2O, with the help of the sun. There's my little sun. Can produce the glucose molecule as well as the oxygens. And you can see the two reactions are direct reverse, which would mean that you have to feeding this some energy along the way. That's where the sun comes into play. The sun can facilitate the ATP needs to do this reverse reaction of the respiration reaction. And when you look at a chloroplast, you see that it's got two membranes. And you got these stacks. Again, there's an outer membrane, there's an inner membrane, and there's the stacks. One single one of these, one single piece, if you will, is called a thylakoid. The entire combination, though, of many thylakoids bound together is called grana, or granum. Many would be grana. And the middle part here is called the stroma. Yes, ma'am?
Why don't the chloroplasts seem to maximize their surface area?
The important reaction in the mitochondria occurs right along this cristae, right along the inner membrane. The important reaction here, in the case of a chloroplast, occurs at the thylakoid and the thylakoid membrane. So the way that it works out, or the best way to have this, is to have the maximization of surface area occur at this location more so than at this location, which is a direct opposite between that and the mitochondria.
What's the stroma again?
The stroma is just the area that is right through here. It's like the matrix is to mitochondria, stroma is to chloroplast. Hmm? Now, there's some features of the two that we needed to know. Before we do that, though, I think it's important we know who has them. So, all eukaryotes would have mitochondria, including plants. Let's get away from the idea that plants only have chloroplasts. They have both. Chloroplasts, though, found in all plants and some of the protists. Fungi, they don't have chloroplasts. Even though they look like a plant, a mushroom looks kind of like it could be plantish, growing out of the ground. No chloroplasts, no sun requirement. But all eukaryotes would have a mitochondria. So that explains how like things work. When we need to eat, we don't go sit out in the sun. We just go to McDonald's or wherever else, correct? And we use our mitochondria to break down the food that we encounter, forming the ATP so that we can do all of our activities, whether that be scratching your eye or whatever else, right? Chloroplasts, though, in plants, what plants have to do is they have to absorb sunlight to do this reaction. They produce sugar, and do you know what they have to do with the sugar then? They have to take it to their mitochondria and do what we do. Plants are on like double duty. They have to make their own food and then eat it, whereas we just go around eating the food of plants. So not only, if you wanted to view it in so many ways, we are parasites of plants, right? We're kind of a parasite of plants. But plants are kind of parasitic to themselves because they generate food only to have to turn around and eat it. And then they have us eating the plants as well. So they kind of have two things against them. That would start to explain why when you look at food pyramids, and if you remember from back in the day, food pyramids a lot of times look like this, and you have your plants on bottom, and you have like you on a higher level above that. There's more plants than there are more of the cows that eat them, and then there's got to be less humans than there are cows, and you get smaller and smaller as you go up. That would explain a large reason why. Cool. Now let's talk about some commonalities between them, some things that they both have together. Some of the things that they have together that we need to put some focus on. They have their own DNA, their own ribosomes, and their own proteins. That seems weird because when we said DNA back in the day, like yesterday, I said DNA and you said nucleus. That's where it's at. Or nucleoid region if you're a prokaryote. But here you see that they have their own DNA. That seems weird because this is supposed to be an organelle that exists only with the rest of the cell. But you're seeing that this has its own DNA. And here's a one-up on that. That own DNA that we're talking about having in both a mitochondria and a chloroplast, it's circular. Do you know who else has a circular DNA? Bacteria. Bacteria have circular DNA.
And why is that?
That's a good point. Mitochondria and chloroplasts, if you start looking at them, they look just like a bacteria. So you have almost these vital structures of eukaryotic cells, and hopefully you've got, at least out of today, that these two guys, vastly important. Vastly important. But they look like bacteria living inside of your cell. Now, you're born with these. You make these along the way, so they certainly can't be bacteria because you're able to generate them. But just because they're not bacteria doesn't mean that they weren't generated from bacteria.
Can I ask a question?
Yes.
So the DNA that they have, is that different from the DNA in the nucleus?
It is different, absolutely. This DNA that you're seeing in both of these guys actually looks a lot like DNA of a bacteria. And so this actually points to something called the endosymbiotic theory, which is a really, really, really important like fact to mention or make. What this endosymbiotic theory states is that once upon a time, eukaryotic cells didn't have these guys. And what happened magically, if you want to, one day about 1.8 billion years ago, was a bacteria that looks like the present-day mitochondria or present-day chloroplast fused with a primitive eukaryotic cell. And now the eukaryote had mitochondrial bacteria in it, essentially. And over time, the bacteria stopped living on their own. Take apart the word. What does endo mean? Inside, within. Symbiotic. This is an early biological term from like 4 years ago. Something about life, it works together. So inside, they work together. Imagine, if you will, that you have a bacteria right here, or I'm sorry, a primitive eukaryotic cell right here. Bacteria that does something very vital walks into it. Does that cell benefit from this new bacteria being inside of it that's a benefit? Absolutely. It's a benefit. So they're going to stay together. And the next thing you know, it's a part of the system. You knew mitochondria were in your cells at the beginning of the day, and you knew chloroplasts were something that you found in plants. And the assumption was that they've just always been there. And what I'm trying to get us to see is that they are there and they are reproduced there. You don't reabsorb new mitochondria each time you make a new cell. But once upon a time, a mitochondria was basically a bacteria that fused with a primitive eukaryotic cell, and they started working together, and they've been working together ever since. Now, then you might say, well, can't that mean that these guys could live on their own again? They can live for a little while on their own, but not that long. It's been 1.8 billion years, and they've started to rely on each other a lot, or they've started relying on the rest of the cell a whole lot, which means that these guys can't exist solely on their own for a long period of time, but they can last for a little while.
How long is a little while?
A couple of days. We'll do a lab where we isolate chloroplasts, and we'll see that they still function without the rest of the cell.
But they can't reproduce by themselves?
They can't reproduce by themselves long term, but when they're inside of a cell, and it's time to make another chloroplast for whatever reason, chloroplasts beget other chloroplasts. But they can't do that, beget other chloroplasts, when they're on their own. They need the rest of the cell to tell them to do that.
Okay.
Cool? So it's kind of a cell within a cell.
Fun fact.
It's always a test question on the AP exam. It's like a guarantee.
Wait,
Some-
repeat the sentence.
That's a guaranteed test question on the AP exam.
The one before
That was like an entire paragraph. Do you want me to say the entire paragraph? So like endosymbiotic theory is where one cell absorbed another cell back in the day, and instead of being two independent entities, they work together. It's like, imagine that, I mean, this is weird, but I can't even give you an example. Imagine that you, I don't know, swallowed a little bug and it stayed inside of your tummy, and the next thing you know, you've got superpowers because of it, and now y'all stay together, and the rest of the human race from this point onward has that little bug inside of it too. That's science. That's sci-fi, is it not? But that's kind of what happened here, just with cells.
Superman?
What?
You got bit by a bug.
Spider.
I've never watched those shows, so I don't know. But we okay with this? What do we think? Pretty manageable, doable? Yeah? Okay.